Good afternoon and welcome to RSSL's webinar on cleaning validation. This is the second of 3 sessions that we're running on this topic, and today's webinar will focus on cleaning validation and analytical perspective. My colleague Steve Alley will be delivering the webinar this afternoon and I'll be here to support him. My name's Lisa Hallwood and I'm the Senior Account Manager within our commercial team at RSSL. Steve Alley is one of our technical specialists within our pharmaceutical development team, and his experience lies in many years of working within early and late phase drug development, and more recently in supporting our customers with their cleaning validation requirements. If you have any questions throughout the presentation, please send a short message in the chat box on the right-hand side of your screen. We'll do our best to answer these questions at the end of the session. So now that we've got the admin aspects out of the way, let's talk about the webinar, and I'll talk a little bit about RSSL so that you can understand why we feel we're able to share our knowledge around cleaning validations with you. RSSL was established in 1987 and has been supporting a number of industries during this time, including food, healthcare and pharmaceuticals on a global basis. We currently work with 20 of the top global companies, as well as small startups and SMEs. Our MHRA and FDA inspected facility in Reading is where our team are based, and our technical expertise covers microbiology, chemistry, physical sciences, and biochemistry. RSSL have been working in the pharmaceutical sector for more than 25 years, and our multidisciplinary teams support our customers from early phase development all the way through to finished product QC analysis. For some of our clients, we'll undertake all of their product development and testing, and for others, they might use us for more specific methodology where they perhaps don't have their own expertise internally, or perhaps it's equipment that they're lacking. Our unique emergency response service ensures priority access to our laboratories and scientists when you're having a crisis or time-critical situations internally. This could be such as resolving customer complaints or dealing with manufacturing issues. In supporting the pharmaceutical industry, RSSL has developed a breadth of capabilities and experience across a number of different areas. These can range from steriles or topicals all the way through to oral dosage forms. We support our customers with either their own methodology, and we regularly work to pharmacopoeia monographs, the most typically being the USP and EP, through to those that are a little bit more diverse or unusual, such as the Chinese, Japanese and Brazilian pharmacopoeias, where our customers are moving into different geographical regions worldwide. RSSL likes to partner with our customers. We like to better understand what your needs are and then refine the project scope to match. We like to keep things as simple as possible, ensuring that the regulatory requirements have been met. This is especially relevant when we look at our cleaning validation projects and our approach to those. I'll hand over to my colleague Steve now, and he'll present the technical content of this webinar and tell us a little bit more about cleaning validation and his analytical perspective.
Thanks very much, Lisa, and good afternoon. Hopefully, at least some of you will have attended the previous webinar that Brian gave on the manufacturing side of this. And what I'd like to just cover before we head on to the slides is that really we might be giving 3 webinars here, but the overall process should be seen as a single entity with a suitable level of collaboration between the various disciplines, and we'll cover that as we go through the slides today. So the first slide here is just outlining the programme of activities this afternoon. So we'll be starting with general analytical considerations, moving on to look at the most commonly used sampling techniques in the factory environment. So we'll be looking at swab samples and then rinse sampling, looking at the use of recovery experiments in assessing methods for suitability, looking at the type of method used, whether that be a specific method, i.e. targeted, or whether we look at more non-specific approach. We're moving on to look at the impact of swab stability data in justifying samples for transport situations. So collaboration is a key part of the overall process, and we need a complete link up here of all of the given expert individuals on the team, and it's really important that you pick the right team for the job. Generally speaking, we would be looking at getting representatives from engineering, from manufacturing, analytical, and microbiology where needed to actually conduct that first plant visit as a team, so you can actually share that knowledge and add valuable input to the process. This not only allows to identify any key problems up front, but also hopefully produces the overall timescale for the process of the cleaning validation. The last point really on here is to make sure that you do bear in mind that these activities do take a significant amount of time, and particularly for analytical activities, you need to be considering these upfront alongside the manufacturing plant walk-around inspection. You know, we're looking at particularly difficult matrices in some of these things. So multiple analytes, you need to be getting a real feel for what is needed up front, so you can tailor your review of literature methods, for example, to ensure that you have a method that's fit for purpose and you have robust analytics for the actual routine testing. So this next slide is really covering some of the key points that really you need to be mentally considering before embarking on your method development program. As we put up before, you need to be carefully thinking throughout your plant walk around, you know, what analytes we need to analyze here. What are the high risk components of this process? How toxic are the analytes? Ultimately, you can have processes where you're looking at 2 or 3 different substances, and you have to then think, do we want one method? Do we want a single method for each active? And that will come down a lot of the time to the types of chemistries you're looking at, and the solubilities, of course, because different chemistries will impart different solubility characteristics. Also, the plant itself will have a number of different surfaces, so your product will come into contact with a whole host of different surfaces across the factory, and you need to be looking up front at these. Which of those surfaces are going to be most in contact with the product? And ultimately, you need to be assessing these in your development program. It's no good just going for one particular surface just for the sake of it. You need to actually be doing a proper evaluation of those surfaces. In terms of analytical methods, you've got several options available to you. Very often, you would have an existing method that you can effectively refine to suit your requirements for cleaning validation. So it may be you have an existing assay method, for example. This method could potentially be curtailed in terms of runtime, and to ensure that you can get the high throughput of samples that's often needed for these processes. Obviously, with residue limits, you will be identifying for each of your analytes a risk-based and tox-based limit that you need to apply to the analyte of interest. So you will have an acceptance limit. Depending on the sensitivity required, you may struggle with certain techniques to achieve that. So again, early stage, bear in mind the sensitivity you're requiring. That will dictate potentially the type of technique you need to use. And finally, specificity, and we're talking here about any of the matrix components that may interfere with your analyte peak of interest. This can come from the swabbing solvent itself. It could come from the swab material, for example. You will get shedding of swab material, and depending on how you're analysing your sample, you may have a less or greater level of interference. So measurement, really, although measurement can be seen as one activity, there's various factors that can input into the success of the measurement of your analyte or analytes of interest. The very first aspect of this that we need to look at is the actual extraction of the active from your surface material. This clearly in itself can pose quite an issue, because depending on your surface characteristics, you can have significant challenges with the extraction of certain components. So for example, if you've got a particularly lipophilic substance, for example, then trying to get it from a rubber surface can be quite challenging. In fact, any substance from rubber surfaces or silicon surfaces where you've got quite a lot of micropores in the material can lead to quite difficult situations when trying to swab from those surfaces. And that leads into the recovery there. So anything you can do to maximize your recovery, and that will come into the swab technique itself, needs to be assessed early on. And quantification, as we say, that will be looking at the type of method you're going to be using and effectively optimizing your solution concentrations and injection volumes, detection wavelengths, and so forth to obtain a method that's suitable for use. So just a little bit about the types of technique available for the cleaning validation purposes. Generally speaking, the main two that you'll come across will be swab and rinse samples. I've got a table here that kind of highlights the pros and cons of each technique. Swabbing typically would be the preferred route, since you can measure directly from a surface. It's a very adaptable technique. The only drawbacks, as I mentioned before, you can potentially get interference from any fibres that might come shed from the swab head itself. It's not particularly useful if you've got very difficult to reach surfaces, or if you're looking behind pipework, for example, in intricate areas, then swabbing really doesn't lend itself to that. And in those sort of cases, you may want to consider a rinse sample. Again, rinse samples, it's a fairly easy technique to perform, and it can be quite quick to do, but you really don't get as much information on the actual level of contamination that you would expect to be seeing with swabs. However, you can justify it in certain instances. It's all about having a body of data to support your approach taken, and this will be scrutinised by the regulatory authorities. So again, just a recap. This is one of the most widely used techniques. The overall principle is to wet the swab itself with the chosen solvent, and choosing a solvent is something that you'll be doing through the development. You'll obviously do a screening test to see exactly what solvents are suitable for your analyte of choice. Also bearing in mind that the solvent chosen will have to be used in the factory, not just in your lab work. So something has to be, the characteristics of that solvent cannot impact on the equipment train, anything in the manufacturing plant that may degrade, for example, rubbers and other surfaces. And a final point here, which I think Brian covered quite extensively in the last webinar, actually, the results that you obtain from swabbing are so dependent on the training of the individual. People think it's an easy part of the work, but swabbing itself can take weeks and weeks of practice, and there's particular exercises you need to do to refresh your skills as well, and you need to be doing it fairly frequently to ensure that you are consistently swabbing and having results that are actually meaningful to any contamination that resides on the surfaces in the various areas of the factory. So for most method developments, I think very often the parameters of the instruments seem to be optimized over anything such as the sample preparation. And I think the same for cleaning validation. Very often there's a lack of attention given to the actual swabbing itself. It's absolutely pivotal to the work you do to ensure that you can demonstrate that the material you're looking for can be suitably recovered at your residue limits. We do this by spiking onto coupons, and these coupons have to represent the surfaces that you've identified in your risk assessments and your plant walk around. So where Brian was talking about cleaning validation master plans and protocols, all of this will be detailed in there, and your analytical work should be reflecting exactly which surfaces you would be assessing in the factory itself. So ideally, or primarily, you should be spiking at the nominal residue limit and quantifying your results versus on the amount you spiked on. So you'll get a % recovery effectively. Ideally, you should be doing this at 50 to 150% of your swab limit, but obviously 100%, 6 replicates, 100% in your development would provide a good starting point for each of the surfaces, not just one, but you do it for each surface. So just to try and make it a little bit more visual, very visual. So you'd start off with your spiking solution. So let's for example suggest that our acceptable limit was 10 micrograms per swab. You would have a coupon of a size. Normally we look at a 10 by 10 centimeter squared coupon. You would spike on an amount of the spike solution to give you 10 micrograms on that surface. You would then leave the coupon to either dry naturally or you can artificially dry it. Following the drying, you would be using a given technique to swab the residues from the plates, and those techniques should be documented SOPs. And again, as I say, there's training required before an analyst can go ahead and do this type of work, both in the lab or in the manufacturing plant itself. The swab heads, once they've been used, are then cut and placed into suitable containers, in this case just some transport tubes, topped up with appropriate volume of solvent, and then analysed using the technique of interest. A bit of a video here as well. This is really just in the lab, just showing effectively the intricacy of the swabbing technique itself. And for this, we've got a number of different surface types. So first of all, we're just highlighting these. So we've got some polycarbonates, we've got stainless steel, we've got glass, PTFE. Okay, and we're just going to start by moistening the swab material in the solvent. And very carefully, using the procedure in place, keeping the swab head as flat as possible, you draw it across the surface material itself in a very regular pattern to ensure that you're capturing any residue right up to the sides of the coupon. And it really is important to keep that swab head as flat as you can to ensure maximum surface area coverage. The plate is then turned and the swabbing repeated. Again, the purpose of this is to ensure you capture any material that may have been present after the first swab. Okay, so the swab is now broken and placed into, in this case, a volumetric flask, and that will be topped up with solvent to the mark. Okay, so having looked at swabbing, the second area that we're going to just have a sort of discussion on it is rinse sampling. And as I said before, this isn't something you'd use as a priority technique. This would be something, if you haven't got a suitable area to swab, you perhaps look at rinse sampling, and it is more for the sort of intricate areas of the plant, and it does only give you a crude measure of the contamination. So in a lab scenario, when you're performing this type of work, you need to have a very careful think about the way in which you're replicating the factory scenario. So if you're going into a factory and you're going to be rinsing a surface, then case one may be your approach in the lab, where you'd actually spike on again onto a suitable surface which replicates that used in the factory, and you'd be using a receptacle to catch the flow from that surface, and then diluting accordingly to give you the relevant solution concentration. In other cases, whereby you are rinsing, for example, a vessel, you would need to adapt the approach you take in the method development work to replicate that. So in this case, we would take a suitable sample of the surface of interest, and we'd actually immerse that completely into a flask with an appropriate volume of solvent, and actually agitate that, for example, using a lab shaker. And that would give you a very, very good match for the actual situation in the plant. So I tried here just to give a sort of a fairly simple worked example of how you would calculate your spike level in a lab situation and how you would achieve a particular concentration in solution. So if, for example, in this case, we've calculated previously, we have a maximum carryover allowance of 5 micrograms per square centimetre. We can calculate as a whole vessel how much that would be. So if our vessel size is 30,000 then we centimetre squared, we can then just multiply that up by the original carryover, which is 5, and the total carryover allowed for the vessel will be 150,000 micrograms. We can then calculate what this would represent in terms of a concentration. So this is your acceptable concentration limit. Again, so you're taking your total vessel amount, which is 150,000 micrograms, and you're dividing that by the vessel volume, and that gives you a figure of 0.5 micrograms per mil. So to achieve that, what you'd be doing here, you'd be taking again your initial carryover of 5 micrograms per centimetre squared, multiplying that by your coupon size in this case, which is 25, which means you spike 125 micrograms onto your plate. And then from there, to achieve a 0.5 microgram per mil, you just dilute that into 250 mils of solvent. It's a very quick overview, but it's just trying to talk you through the thought process in determining those levels. So I think, again, the point with this next slide is to demonstrate that you can't readily rely on rinse samples to tell you the whole story. You can't really test, for example, if you were washing up, trying to test the water after you've washed up for the cleanliness of your plates is not going to give you the full story. Clearly, it's not going to tell you how clean those plates are. It'll give you an indication, but it won't give you an absolute number. Okay. So types of methods. This is a topic that's been covered quite a lot actually in recent years with regulatory involvement, and really depends what situation you've got in terms of where are you in your product life cycle. How long have you been doing your cleaning validation and subsequent cleaning verification for? Normally, non-specific methods are frowned upon because they don't give you enough information. However, as I said earlier, non-specific is a method that is not targeted to a given analyte. And in that sense, running a non-specific method can be quite advantageous, as it gives you a real flavour of the gross contamination you might have in your plant, not just your identified analytes. However, you really need to be going down, if you've got a fairly new process, you need to be identifying individual analytes, particularly if you've got particularly toxic materials with low limits. You need to be first getting on handle and controlling your process. And once you've controlled your process and you can consistently demonstrate you're achieving less than the residue limits, at that point you may decide, well, if my analyte can be detected, for example, by total organic carbon, then we could move to that, since it's a much cheaper technique to run. It's going to mean your downtimes in the factory are a lot less, and overall going to be a more efficient technique to use. But you have to generate a lot of data to allow you to take that approach, and it's all about body of data and effectively presenting as much as you can to the regulators to justify your position. The specific techniques, I think probably for most people, HPLC is the preferred technique. Obviously sensitivity can be often an issue. Sometimes you can use LCMS, but predominantly LCUV would be the chosen technique. Although we have got an example, there's a factory tour I did a few months ago where part of the process was actually using a brass bristled brush to actually clean sieves in as part of that processing. And what we decided in that case was to actually employ atomic absorption, see if we could capture, see if any metallic material had been lost. So in that case, atomic absorption is a very good technique to do that. So just to watch out, make sure you think about the types of things you're analysing, what you're looking for, what limits you're looking at, to try and really hone in on the most appropriate technique to use. Again, this swab stability is something increasingly that we have customers coming to us to conduct remedial work, and where you are working remotely, for example, with a contract lab or the supplier, you need to be confident that any transport of materials is supported by a suitable package of stability data. You want to make sure that the swabs you take, when analysed, will give you results that would actually be realistic of what was present originally. It's all very well saying, you know, we've sent these swabs out for analysis, and well, you know, the limits come back and it's below the acceptance limit. Great, all passed. Have you got the body of data to confirm to yourself and your regulators that those low limits were indeed due to a realistic swab taken in the factory, or are those low limits due to degradation of the active or actives since the swabbing occurred? And you need to therefore be mindful in the development. It's always good just to, alongside your development, store some samples in swab. Once you've got your swab solvent and your volume sorted out, make sure you start gathering that data so you can actually present that data in your report and effectively your method to say that I've got stability, for example, for 7 days at ambient. Okay, I think just moving on to really a sort of summary here. I think, you know, looking at the presentation we had last time with, you know, we were looking at the manufacturing side. So walking the plant is not only important for manufacturing, it's important for the other members of the team. Collaboration is absolutely key as early on as possible to try and effectively concertina the process as much as possible. Swab training, again, we're talking here about not only in the manufacturing plant, but we're talking about between your analysts in the lab to ensure that you're getting repeatable, consistent results. And all of these will lead to much higher chance of getting robust analytics. So whilst this is quite a sort of overview, obviously we have been working on a significant number of projects, and we are more than happy, as I say, and we will in a minute take some questions that may have arisen. So Lisa, if I can hand over to you.
Thanks, Steve, and thank you for the overview. I think that was really useful. As Steve's mentioned, we've had some questions come in during the session, so we'll spend some time now just going through those with you. Hopefully we'll capture their meaning as they're intended, but please let us know if we've misread them or misinterpreted them. So Steve, one of the first questions that came through was, how similar do the swab surfaces used for the analytical method development need to be relative to the manufacturing plant?
The short answer is they have to be the same. Very often, you know, when you've got an older factory, for example, it can be very difficult to find the specifications for all of your surfaces of interest, and that's another story. That has to be a risk-based approach. So if you can't get specifications, you have to do your best to identify what those surfaces are. For where you have a factory that, you know, when your factory is put together, you should have specifications for all of the component materials that those products are going to come into contact with. And where you've done your cleaning validation protocol, you need to be ensuring that all of those materials identified are assessed in the method development, because they're all going to have different recoveries, they're all going to potentially have different interferences if you get extraction from any of those surfaces. And you can order coupons from you know, particular companies, different sizes, different materials. So yes, the answer is you have to mimic exactly what's in that plant.
Perfect, thanks, Steve. One of the other questions we've had come through was, what approach should be taken if low recoveries are obtained during the recovery experiments?
Quite an open question. There's lots of scenarios we can look at here, and I think recoveries in general, we in cleaning validation have a process of using correction factors. Now this would be a case, so for example, and let's just take a recovery of more than 50%. So long as you can show that you're getting a consistent recovery, so your precision between your replicates is good, and you can do that over several days and several HPLC systems, for example, whatever technique you're using, then that gives you grounds to say, right, well, we know that with this technique on this surface, we're only going to recover this amount, therefore we can put a correction factor in place each time we analyze that, that's fine. If you're getting less than 50%, there's two things you can do. Firstly, you can adapt your swabbing technique itself. You could go to double swabbing, and that could ensure that you maybe capture anything that you've missed on the first round of swabbing. Alternatively, Or additionally, actually, you can actually go back to the manufacturing plant and you can identify how much of the manufacturing train, the particular surface of interest that's giving you problems covers. Because if it's a very low percentage in terms of the overall manufacturing plant, you've got very good grounds there to actually risk assess. So, well, actually, yes, we're getting low recoveries, but in the grand scheme of things, it's only touching, say, 1% of the overall surface. You probably wouldn't worry quite so much about it. And obviously, you need to think about the toxicity of the compound as well, but you need to be doing that risk assessment as well. That can give you a much stronger body of data.
Thanks, Steve. We've picked out one final question for today's session. Those that we haven't got to, we'll follow up separately. Steve, do you have any thoughts on the ability of an analytical method to quantify below the calculated acceptable residual limit?
Sure. Again, I think from an ethical standpoint, we're here to ensure the patient's safety. Whilst you can calculate a given limit, if your technique is capable of going a bit lower, what's the problem? I think from a personal perspective, we've in the past we've gone below because we can. Important thing there is just to make sure that you fully evaluate the limit, because you don't want to be in a situation where on one occasion you've achieved, say, half of the limit you've assigned as your acceptable limit. If you can't do it on a second day on a second system, you're going to put yourself into a lot of hot water. If you've claimed that as your limit, you need to be very careful and mindful about the amount of data you have for that. So yes, I think, yeah, if you can go below, but it's not the be all and end all. Don't let it drive your development. You just need to be ensuring that the limits you've calculated and risk assess you can achieve.
Great, thanks, Steve. That brings us to the end of our webinar for today. We hope you found it useful, and we do really appreciate the questions that have been put into the session, so thank you for that. As I said, we've not managed to answer all of the questions today, so what Steve and I'll do is follow up with you separately. Steve's contact details are on the screen, so feel free to get in touch with him directly if you have any questions that occur to you after today as well. The next webinar in this three-part series is taking place in July, and it's going to focus on the microbiological considerations for your cleaning validations. So hopefully you'll join us for that session as well and bring some challenging questions for our team on that one. So on behalf of the team, I'd like to thank you for listening today. As I say, please don't hesitate to contact us if you've got any questions. We look forward to joining you on the next webinar. Thanks very much for your time. Bye.